Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Think about your daily routine. That sugar-free soda you might have with lunch or the protein bar after a workout, maybe even some toothpaste. You've likely seen a name pop up again and again, erythritol. It promises, no sugar spikes, almost 0 calories.
Yeah.
Sounds perfect for everything from keto diets to managing blood sugar. Like a total win.
Right. Seems like it on the surface.
But what if this widely used sweetener isn't quite the guilt-free miracle we thought it was? Today, we're doing a deep dive into some new research. It's kind of cracking open a surprising side of erythritol. And this one's not about weight gain or tooth decay, but something potentially much more critical for your brain and heart. To guide us, we've pulled together some really fascinating sources.
That's right. We're drawing heavily from a recently published scientific paper. It's in the Journal of Applied Physiology, authored by Auburn Berry and colleagues, and led by Professor Christopher D'Souza from the University of Colorado Boulder. This study gives us some crucial insights into the mechanisms, how it actually works at a cellular level. It builds on earlier research that sort of hinted at these connections.
Okay.
And we also have some valuable context from a health news article from earth.com, which helps translate the scientific findings into a broader picture for you.
Okay, so let's unpack this then. For those who might not know, what exactly is erythritol?
Right. So essentially, it's a sugar alcohol. Now, you can find trace amounts of it naturally in some fruits, sure. But the vast majority that we consume, it's mass-produced, usually from corn or sometimes wheat starch. And its big selling points, like you said, are that sweet taste without significant calories. And crucially, it doesn't cause those blood glucose spikes like regular sugar.
And that's exactly why it's become so popular, isn't it? I mean, it sounds like the perfect solution for reducing calories, managing blood sugar.
Absolutely. And, what's really interesting here is how it became such a go-to. Unlike some other sugar alcohols, it tends to cause fewer digestive issues. That's because our bodies absorb most of it before it reaches the colon. But, this efficient absorption also means it gets into your bloodstream pretty quickly. And importantly, the new research indicates it can actually cross into the brain.
Okay, that's where it gets really interesting, because our sources point out that even before this current study, there were already some, let's say, unsettling concerns bubbling up.
Yeah, there were hints.
There was one large epidemiological study. It tracked over 4,000 people, I think, across the U.S. and Europe.
That's the one.
And it found something quite striking. People with high blood levels of erythritol had a higher incidence of strokes and heart attacks within 3 years. And this was regardless of other health factors.
Exactly. But that earlier finding was largely correlational. It showed a link, a connection, but not how or why.
Right, it didn't explain the mechanism.
Precisely. So this new research directly addresses that how. The goal was to determine in a controlled lab setting in vitro, using cells in a dish, what effect erythritol has on human brain microvascular endothelial cells.
Okay, break that down a bit. Human brain microvascular endothelial cells.
Yeah, it's a mouthful.
Yeah.
But basically, these are the specialized cells that line the tiniest blood vessels in your brain.
Got it. The really small ones.
The really small ones. And they're absolutely critical for maintaining healthy blood flow, controlling inflammation, and preventing dangerous clots from forming.
Okay, so super important cells. How exactly did they test this connection in the lab? What was the method?
So the researchers took these human brain blood vessel cells and exposed them to erythritol for just 3 hours.
Only 3 hours?
Yep, just 3 hours. And the amount they used was 6 millimolar.
Which means?
Which is roughly equivalent to what you'd typically get from drinking a single sugar-free beverage that contains about 30 grams of erythritol. They wanted to mimic a kind of real-world single exposure.
Okay, so a realistic dose, short exposure. What was the very first thing they noticed?
Immediate stress. That was the first finding.
Stress on the cells.
Exactly. The cells were basically flooded with unstable molecules called reactive oxygen species. You might know them as free radicals.
Right. Those aren't good.
No, they're known to damage proteins, DNA, cell membranes. It's like a direct assault on the cell's health. Now, In response, the cell's own natural antioxidant defenses, their internal cleanup crew, you could say, tried to ramp up. They tried to fight back.
But
But crucially, it wasn't enough. They measured a significant 75% increase in these damaging reactive oxygen species, even with the defenses trying to compensate.
wow, a 75% increase. So the cells are immediately under attack. What happened next? What was the next domino to fall? Did it affect blood flow?
It did. That short exposure, just 3 hours, changed how the cells produced nitric oxide.
Nitric oxide.
Yeah.
That's the stuff that helps blood vessels relax, right?
Exactly. Think of it as the body's natural signal for vessels to widen, to loosen up, and let blood flow freely. A vasodilator. What happened was the enzyme responsible for making nitric oxide, it's called endothelial nitric oxide synthase, well, it just stopped doing its job properly. Its activation was messed with.
So less nitric oxide.
About 20% less nitric oxide production, yes, which directly sets the stage for blood vessels to become constricted, less flexible.
Okay, so they're less able to relax. But did anything actively make them tighten up even more?
Yes, that was the next finding. Connecting this to the bigger picture, the very same cells started pumping out more endothelin-1.
And endothelin-1 is?
It's a protein that basically tells blood vessels to constrict, to tighten up. Compared to the normal unexposed cells, the erythritol-exposed ones had a 30% increase in producing this endothelin-1 and also more of its precursor molecule.
So it's double whammy then.
It really is. The vessels lose their ability to widen effectively because of the reduced nitric oxide, and they get stronger signals telling them to squeeze tighter because of the increased endothelin-1.
Okay, constricted vessels.
Yeah,
That sounds like bad news for blood flow, especially in the brain.
definitely.
And you mentioned clots earlier. What about those? Because that seems critical for stroke risk.
Absolutely critical. So your body needs to form clots, obviously, when you bleed, but it also needs a system to break down any unnecessary clots that might form inside blood vessels.
Right, to prevent blockages.
Exactly. And one of the body's key tools for breaking down clots is something called tissue-type plasminogen activator. Let's just call it TPA.
TPA, okay. The clot buster.
The natural clot buster, yeah.
Yeah.
Normally, when there's a clotting signal present, they use thrombin in the experiment. These endothelial cells ramp up their release of TPA by about 25%. It's a protective response.
Makes sense.
But in the cells that had been exposed to erythritol, that response was completely blunted, gone.
So clots could form.
Clots could form, and the cells just didn't react properly to release the tools needed to break them down effectively.
Wow. That seems incredibly relevant to stroke risk.
It does, and the first author of the paper, Auburn Berry, put it very directly. She said, and I'm quoting here, "Big picture, if your vessels are more constricted and your ability to break down blood clots is lowered, your risk of stroke goes up."
A direct link.
She continued, "Our research demonstrates not only that, but how erythritol has the potential to increase stroke risk. It shows a plausible biological how."
That's really powerful evidence at the cellular level. But like with any single study, it's important we talk about the limitations, right? What doesn't this specific study tell us?
That's a really crucial point to make. First off, remember, this was an in vitro study.
Meaning in a dish, not in people.
Exactly. It was conducted on isolated human cells in a lab, not directly in living, breathing humans. Now, these specific human brain blood vessel cells, they are a well-established and validated model for studying how the brain's microvasculature works. But still, we can't jump to definitive clinical conclusions solely from this type of study.
Okay, that's important context. What else?
Second, the study focused on acute short-term exposure, just those 3 hours.
Right, and that brings up the obvious question.
Yeah.
What happens when cells face erythritol, not just for 3 hours one time, but maybe every day, multiple times a day, which is how many people might consume it, right?
Yeah.
In drinks, snacks.
Exactly. And the paper itself acknowledges this limitation. They state that considering people often consume drinks with these non-nutritive sweeteners multiple times a day, it's reasonable to, hypothesize that chronic elevated levels of erythritol circulating in the bloodstream could lead to persistent, long-lasting negative effects on these vessel cells.
So more research needed on long-term use.
Definitely. Additional clinical studies looking specifically at long-term health effects in people are certainly warranted based on these mechanistic findings.
Okay, so putting it all together, what does this mean for you, the listener, making daily choices? Given these findings, what's the takeaway?
Well, Professor Christopher D'Souza, the senior researcher on the study, he pointed out something important. He said, the Colorado team didn't just observe changes. They showed exactly how the sweetener weakens brain vessel cells in ways that match the stroke patterns found in people.
So connecting the lab findings to the earlier population studies.
Right, providing that potential mechanistic link. Given that, Professor D'Souza suggests, and again I'm quoting, it would be prudent for people to monitor their consumption of non-nutrient sweeteners, such as this one.
Prudent to monitor.
Yeah.
Not necessarily eliminate, but be aware.
Exactly. This isn't about causing fear, it's about being informed. For you listening, it might mean just taking a closer look at ingredient labels next time you shop.
Or maybe exploring other unsweetened options, or different types of sweeteners perhaps,
Yeah.
though they all have their own profiles.
It's about conscious choice based on the available information.
And this really brings us to a fundamental sort of dilemma, doesn't it? In our efforts to avoid one perceived health risk, like too much sugar,
Yeah.
are we inadvertently exposing ourselves to others we don't fully understand yet?
That's the million-dollar question, isn't it? As we move towards this idea of a sugar-free world, really understanding the full impact, the nuances of all these substitutes becomes absolutely paramount.
We've explored the cellular mechanisms here, the potential risks highlighted by this study, and also the limitations of the current research. What really stands out to you from this deep dive today, and how might this information change your approach to what you consume? Something to think about.
Definitely food for thought.
Well, that's all the time we have for this deep dive into erythritol and its perhaps surprising effects on the blood vessels in your brain. Thank you so much for joining us on this journey to become a bit more well-informed. We really hope this exploration gives you something valuable to consider. Until next time, keep digging, keep learning, and stay curious.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.